Okay, this is Bio1005 Chapter 15, Diversity of Animals. And the animal chapter, animals are unique because they just digest their food within their bodies. They're eukaryotic, they have multiple cells, they're heterotrophic organisms that obtain nutrients by ingestion. So heterotrophic means they eat others. If you break down the word hetero, others, trophic is eating. So some features of animals. Most animals are going to reproduce sexually. You can see here you've got the adult with 2 sets of chromosomes, and then you've got eggs and sperm made by meiosis that have 1 set of chromosomes. The egg and sperm fertilizes, goes through mitosis, and we have this cycle here. There are some exceptions, and those are listed down here at the bottom. There's an interesting phenomenon called parthenogenesis. This is when unfertilized eggs develop into offspring with just the female. They have had no encounters with males. Some insects also, the males are haploid, and it's the queens that are reproducing. So let's see, I do want to point out this picture of the sea star because in our lab we're going to be looking at sea stars, and we're going to be talking about some things like radial and bilateral symmetry. So I just want to show you here a zygote, that's a fertilized egg, then that's going to multiply through mitosis. Then we get this blastula stage and then a gastrula stage. I am not testing you on those are not in your study guide. But then you get a larva and then you get an adult sea star. We're going to talk about radial versus bilateral symmetry pretty soon, but I just want to show you that an early stage of the sea star is bilateral. So most animals are going to develop specialized cells. In this example, we have nerve cells. In this example, even very simple organisms have brains, eyes, intestines. You have a mouth, an anus. So you've got all kinds of specialized cells going on, specialized tissues, nervous, muscle, connective, epithelial. The exception to that is going to be our simplest animal, which are sponges, and they have no true tissues. So if you look at all of the phylum, remember the word phylum, it comes from the classification system where you have domain, kingdom, phylum. So we are looking at the kingdom Animalia here, and this is showing you some breakdowns. And we are only going to be looking at the phyla with the red arrows. So we are going to miss out on a few things because these 9 phyla are kind of the main ones. And we're going to skip rotifers, ribbon worms, and comb jellies. So just so you're aware of that if you ever take a higher zoology class. So let's look at body plan. Now we're finally getting into our study guide. So if you look at the first page of your study guide, you've got radial symmetry and bilateral symmetry. Those words are defined here. I'm not going to pause much because I know with this video you can pause yourself. So I just want, here's the definitions, and then you can see in this picture this thing is not symmetrical at all. That's going to be our sponges. Radial symmetry is when you can slice it any which way and you would get equal halves. So you could slice it that way or that way. Whereas if you look at bilateral symmetry, a goat, you cannot slice it, say, this way across its body because then you'd get a head on one side and a butt on the other. So you can only slice bilateral down one plane, to get equal pieces. And of course, this isn't perfect because if you look at a human, our heart tends to be on our left side. So if somebody sliced us down the middle of our face, through our nose, through our belly button, you wouldn't exactly get equal pieces, but for simpler animals, this is the case. So now we're going to look at tissue layers. This continues with page 1 of the study guide. We've got the word diploblast and triploblast. So this is referring to embryonic development. As the embryo, so you've got the fertilized egg, and then it becomes the embryo. Most animals are going to develop layers of tissues, and those are called germ layers. So radial symmetric animals, those are the ones you can chop any which way from the top down. They're going to develop 2 germ layers, and they are going to be called diploblasts. So you always want to look at the prefixes in your word. Di meaning 2, so a diploblast, 2 germ layers. That's going to be radial symmetrical animals, and those are going to be our simpler animals, the ones that evolved first. Then we have triploblasts, and these are going to be your bilaterally symmetrical animals. They're going to develop 3 germ layers, and we've got that tri right there in the prefix, tri meaning 3. So looking at the picture, the triploblast has indeed 3 different layers, and the diploblast not quite at 3. They just have the 2. So those are your next 2 terms on your study guide. Okay, now we're going to talk about body cavities, and triploblasts can develop what's called a coelom. And this word coelom means a body cavity usually filled with fluid between the body wall and the digestive tract. And you might be thinking, who cares? What is the big deal with these different body cavities? Once you get a more advanced body cavity, then you can start to get more complicated, like organ systems being made. And then you can start getting more efficient processes done in the body. So if we look at no body cavity, this is going to be our triploblast, the flatworm. He's got, if you look, we just have a digestive cavity, and then it's kind of solid tissue layers through his body. So he doesn't really have a body cavity. The digestive tract is not a body cavity. So then as we move to the roundworms, we've got a pseudocoelom. So you've got the hole for the digestive tract, which is different. Then you've got a layer of tissue around that, and then you've got this layer of tissue here. So that's called a pseudocoelom. That is also on your study guide. And then we get a more complicated animal like the annelid. We're looking at 3 different kinds of worms here, and this is a true coelom. So the true coelom means that it's a tissue layer lining the coelom and suspending internal organs. So it's like this mesoderm is surrounding both the digestive tract and the outside layer of tissue. Okay, so that is going to be your true coelom. All right, continuing on with embryonic development, we've got the words protosome and deuterosome on our study guide. So this you can kind of think of with a joke. Animals are divided whether their mouth or their anus develops first. So with the protosomes, the mouth develops first. Examples of protosomes are going to be your arthropods, which includes your insects, mollusks, which include clams, and annelids, which are your like earthworms. So we've got the mouth developing first, and then the anus develops. With deuterosomes, the anus develops first. Okay, and this is what we are. So we are kind of asses because that's what develops first, is our anus. And this includes the chordates and the echinoderms, and the echinoderms is the one that sea stars are in. So if we look at kind of the evolution of early animals, we have talked about the Precambrian time frame being when the Earth first developed, and then oxygen came, and then we had our first eukaryotes form. So this is showing you how animals probably evolved from a colonial, so they get into a colony, flagellated, that means they have the little flagella, protist that lived in Precambrian seas. And you can see these resemble our early stages of embryonic development. And then it would take, remember this took billions of years, reproductive cells forming, and then a little digestive cavity starting to form. And this actually looks very similar to our very simplest animal, the sponge. So these are just some super creepy pictures of fossils that we have found of some animals that lived 542 million years ago. So at the beginning of that Cambrian period, animals just went through a huge diversification, and many of these are extinct now. And can you just look at this and say, oh my God, thank goodness that thing is extinct. It's a little scary. So what we're going to do next is we're going to be looking at the next 3 pages, actually the next 2 pages of our study guide. If you look on that first page, you will see this graphic. Each one of these is one of the 9 phyla that we are going to go into. And for the next exam, this is going to be a case where I want you to give me an example of each phyla and the characteristics that match that phyla. Okay, so these are considered the earliest, and we will be going to the chordates at the end. Now the vertebrates fall into the chordates. So everything you probably think of as being an animal, like something that you see at the zoo, is probably a vertebrate. So we will be talking about those on page 3 of your study guide. So our very simplest animal, if you look on the bottom of page 1, you've got the phylum Porifera. We're just going to call those the sponges. Okay, so the characteristic that you need to know about sponges is going to be that it lacks true tissues. That's its characteristic. And an example is a sponge. So that one should be pretty easy to get on the exam. So if we look a little bit closer at a sponge, it kind of looks like a sack with a bunch of holes in it. It draws water into its digestive area, a central cavity, and then there are these little flagellated cells that catch the food particles, and then the water is blown out. So there's kind of this circle of water. Now during your, we're going to be doing a lab on animals, and that lab is going to ask you, why does a sponge have pores? So I'm kind of giving you the answer right now. A sponge has pores so it can bring food particles into its central cavity, and then these cells are going to catch that food, and that's how it gets its food. Let's move on to the cnidarians. So that's going to be on page 2 of your study guide. This cnidarians, their characteristics, they have 2-layer body tissues, so they're diploblasts. They have radial symmetry, and they have tentacles with stinging cells. So if you need to go back and review what the word diploblast and radial symmetry means, you can go ahead and review that now. But the basic body plan of a cnidarian, it's going to be a sac with a gastrovascular cavity. It has two variations, the polyp, where the tentacles face upwards, and the floating medusa, where the tentacles face downwards. So if we look, this is the polyp. We have the tentacles on the top. This is the medusa form with the tentacles facing down. In your lab, you will be asked to draw this one. It's important to note that these guys, their mouth and their anus is the same hole. Gross, collective gross. You are probably very familiar with, most people call them jellyfish, but jellyfish is not scientifically accurate. They are jellies. If you've seen Finding Nemo, you're probably familiar with sea anemones. So going back to Finding Nemo, which is, a really good source of scientific information, it actually is. The teacher in that movie is a stingray, and no, I'm sorry, I'm doing the wrong. The turtle is talking about taking on the jellies. So I got my Finding Nemo reference wrong, but taking on the jellies means that Nemo's dad went through the jellyfish. All right, more examples of cnidarians. We've got polyps, hydras, sea anemones, and coral animals. So when you see a piece of coral, it kind of looks like a rock, and it's got these tiny holes in it. Each one of those tiny holes at one point had a very small animal in it, like this. Okay, very small. So a coral is a cnidarian, and the piece that we see is rock-like. That is the leftover exoskeleton, or not exoskeleton, but that's the leftovers of their ancestors. Okay, so hint, hint, wink, wink, nudge, nudge. You have to look at some unknown animals in the lab once you've gone through all the phylum, and you have to identify them. So remember that coral are cnidarians. Cnidarians are carnivores, so they use their tentacles with these special stinging cells to capture prey. And as the prey brushes up against it, this whole thing snaps open, discharges the thread, wraps around the prey. The way, if you ever get stuck, if you ever go somewhere and you get hit by a jellyfish, not likely to happen in Colorado, you can lower the pH. So if you pour something acidic, like vinegar, or if you've watched that Friends episode with Joey, you can also use urine, because that's usually a lower pH. You can get the jellyfish to let go. I'm going to stop there. So the first video is under 20 minutes, and we will continue with another video.